9 o'clock. Oh, well, now we started recording. Okay, it's 9 o'clock. Good morning, everybody, and welcome to Third Thursday for May. I'll introduce our presenter in a moment. A couple of housekeeping things. We plan to shift the time for the summer. So June will be the same, will be 9 o'clock on the third Thursday of June, which will be the 15th. Christopher Mark from Vitality will be presenting on soil health and testing. And then for July and August and maybe September, we're going to have a noon to 1 third Thursday, which hopefully will give you VIT people some time to work in the cooler hours and take a nice lunch break and join us and ask questions and get information. So stay tuned for that. Kate will send all that information out to you. Today we have Dr. Tanja Voegel, who's presenting on crown gall disease of grapevines. Dr. Voegel is a research associate at the UBC-O. Her work focuses on Agrobacterium vitis, the causal agent of grapevine crown gall. She obtained her doctoral degree in 2010 from the University of Freiburg, Germany, and UC Davis, where she studied Xylella fastidiosa, the causal agent of Pierce's disease on grapevines. She worked with Dr. Walter Gubler on grapevine trunk diseases and the powdery mildew risk index model. So welcome, Dr. Voegel.
Thank you very much. Thanks everybody for being here this morning. So my presentation is going to be about crown gall disease of grapevines. And I'd like to respectfully acknowledge the Okanagan Nation and their peoples in whose traditional, ancestral, unceded territory UBC-Okanagan is situated. So grapevine crown gall is caused by the bacterium Rhizobium vitis. And there are other names out there. You may have heard Agrobacterium vitis or Biovores 3. So for this presentation, I'm just going to abbreviate as A vitis. So it's important also that it is a bacterium. So it's not a virus or a fungal disease. Those are causing other diseases like trunk diseases for fungal pathogens or virus diseases like red blotch or fan leaf virus. A vitis is the most destructive bacterial pathogen of grapevine, but unfortunately, we don't really have good economic data to support this. There's only one study available from the US that showed that in a 0.4 hectare vineyard, there was a damage of about $47,000 US dollars over a period of 6 years. So we don't, unfortunately, have any data for the Okanagan Valley about that. Crown gall is mostly prevalent in cold climate viticulture areas, but also in other areas. As you can see here on the map, everything in yellow shows areas where crown gall is present. An important thing, though, is that crown gall is host specific to grapes. So practically, that means if you're planting a new vineyard, and in that vineyard, there was only pasture or grassland or fruit trees before, there will not be grapevine crown gall in there because those bacteria are only associated with grapes. So this slide shows many symptoms of crown gall, except one of the slides. So this requires a little bit participation from the audience. So if you all want to have a look at those photos and maybe let me know in the chat function which one you think are not crown gall symptoms. Anybody have any ideas? You may remember that from my last presentation. Oh, number 2. Yeah, and that is right. Number 2 is galling from a nursery. So this is not caused by crown gall bacteria. This is through the grafting process. So these are the regular gall symptoms that we see. That's at the graft union. And these are symptoms that look closely related to water stress. And that is because crown gall does cause, it is water stress basically, because the galls, they girdle the trunk and then less water can flow through the xylem and we end up with symptoms like these. These shows, oh, sorry, do you see the pointer? No. Galls along the trunk. And then this has been very infected. We see galls all the way up into the cordon. And this is how fresh galls look when I inoculate the bacteria artificially in a greenhouse. This is heavily infected with galls, but even in nurseries, like this is a nursery in field, and we can see galls already forming there. And this vine very nicely shows the comparison. So this is a healthy vine on the left, and on the right, we have a heavily diseased vine. So down here is all the galls. They girdle the trunk and no water can flow, and that results in a reduced growth of the plant. And in these vineyards had to be replanted. All of those vines that had to be taken out were crown gall diseased. Some have made it through, so they didn't affect all of them. And then another important symptom is root necrosis, as shown here. And that's important because if roots are necrosed, then other soil pathogens could enter vines through those wounds, like fungal pathogens, for example. So in general, symptoms of crown gall are gall formation on the aerial plant parts. And then fruit ripening is delayed, sugar content of the berries is reduced, and in general, there is a reduced yield and vigor. Shoots grow inferior, and then there is root necrosis. So this is the disease cycle. Let's say you're wanting to establish a new vineyard and you order nursery material from the nursery. You look at it and you think, oh, this looks healthy, I'm going to plant this. However, the bacteria can reside asymptomatically within those planting material. So they are there, but we don't see them. Upon planting, the vine grows, and then any type of injury that can happen to the vine can then start the formation of galls. So injuries can be anything, for example, disbudding, grafting, mechanical injuries through some of the machinery that's being used. But mostly it's freeze injury. So bacteria will move towards the sites where the wound has occurred, and then galls will develop, and bacteria will also distribute throughout the vine. In fall then, or in winter, when the vine becomes dormant, the hypothesis is that the bacteria move into the roots of the vine and overwinter there. And then they're being pushed up in spring again with the xylem sap, and the cycle continues. So this is what's published about the disease cycle, but I added a little information down here. So you see that your vine has symptoms, and you're going to remove this vine, which would be a good idea from your vineyard. However, the bacteria can stay in the roots of those vines in the soil for up to 2 years. So the data so far only suggests that they can stay for up to 2 years. We don't really know what's happening after. So now you have soil where the bacteria are still in there. You're planting new vines into that soil, and bacteria can reinfect the roots from those newly planted vines, and the vines can become sick again. So there are two ways how the bacteria can enter in the vines. So the first one would be through the contaminated nursery material, and the nurseries themselves, they may not even know that the material is contaminated. And then the second way is through contaminated soil that's already in the vineyard. This is a schematic how the tumors form. So here is the bacterium, and this is the plant cell. So upon wounding of the plant cells, the cell will release wound hormones, and those hormones attract the bacteria. So they all move towards the wound. And then the bacteria have something called a TI plasmid, and that's a piece of DNA. And on this plasmid, there is a tDNA, it's called transfer DNA. This transfer DNA is moved into the plant genome, and it's integrated there into the plant cell. The tDNA then codes for the formation of hormones, auxin and cytokinins, and these hormones result in tumor formation. So this is what's making the tumors. It's basically plant cells that are overgrowing. But the tDNA also codes for enzymes that synthesize opines. And opines are basically carbon sources for the bacterium. So the bacterium is really smart. It makes the plant produce its own habitat and food for the bacteria to survive. So here are a few management practices of things that don't work. elimination of the bacterium after it's been established in the vineyard is going to be very, very difficult. And that is because, if you remember the last slide, the DNA is inserted in the grapevines, so it's going to be there. Hot water treatment is something that is being done by many nurseries before cuttings or dormant nursery material is shipped. And it has been shown that 30 minutes in 50 degrees Celsius hot water has been able to reduce the bacterial population, but it wasn't able to eliminate all of the bacteria. But it still may be beneficial, and I'm going to get back to this later in the presentation. So unfortunately, we don't have any systemic chemical control to control grapevine crown gall in the vineyard. So there are many chemical products that work against the bacteria on the plates. For example, there's copper, there are antibiotics. So there's even a study about oregano extracts, and there are many other compounds who control the bacteria, but we have nothing that we can apply at the outside or spray in the vineyard that will then kill the bacteria that are residing systemically inside the plant. There are biocontrols available, but those biocontrols don't work against grapevine crown gall. So all of the biocontrols products that are out there on the market that contain K84, for example, the products Digol or Gallchol, they do not work against grapevine crown gall. Then this is something that we have tried out in our lab, transient heat therapy. And you can see that on the photo here. It's basically this machine that's being towed by a tractor, and it's going through the rows in the vineyards. And these two are fans, and they blow hot air into the vineyard. It's propane fired. So it's about 150 degrees of hot air that's being applied into the canopy. And reports where the machine has been used claim that there was an increase in yield, for example, but there was also a reduction in plant diseases because the heat turned on some plant responses that allowed the plant to fight the pathogens. So we've tried it over 2 years in 3 local vineyards here, and we did not see any effects in yield or improvement of plant performance. And we certainly did not see any effect of heat application to control crown gall. So here are a few management practices that do work. And this slide shows the things that can be done before planting. And everything that's highlighted in yellow are some of the studies that we have done in our lab. And I'm going to expand on those topics later on in the presentation. So early detection is very important, because with early detection, we can say if the vineyard soil is free of bacterium or if the planting material is free of the bacteria. And there are 2 methods available that are cited here. We do have the option now to develop a vitis-free propagation material, and that's being done through shoot tip tissue culture. So that method works. However, those vines are then planted or propagated in vineyards and have to be grown. And the problem is that some of the contamination is coming in later into those vines that have been initially crown gall free. And there's also the option to choose less susceptible varieties. And I'm saying less susceptible here. I'm not saying resistant, because there are no varieties that are completely resistant. All of them will have crown gall or can have crown gall to some degree, but the most susceptible are Vitis vinifera, and then Riparia, Nebraska, or hybrids are somewhat less susceptible. The same for rootstocks, 3309, 10114, and Riparia glor and Teleki 5C seem to be less susceptible in studies that have been published. However, those are most of the rootstocks that we have here in the Okanagan, and many of those rootstocks did develop crown gall. So we think that there is maybe an interaction between some of the local pathogen strains that we have here and the rootstocks, and that we can't generalize this for all of the locations. The cyan choose hardy cultivars. So for example, Pinot, Riesling, Cabernet, or Chardonnay. And this next point may be obvious, but if there are galls in a vine, do not propagate from those gall vines, because the bacteria can be anywhere, and it's likely that any cuttings you make from those vines will also have the bacteria present. So site selection is something that's really important. And you have to make sure that there's good air and water drainage and avoid any low-lying regions and avoid anything that can cause frost injuries, so any areas that are prone to that. Nematodes play a big role in evitis too, and we have done a project together with Dr. Tom Forge. Nematodes feed on grapevine or parasitize grapevine roots, and those wounds can then serve as entry points of the bacteria. In general, it would be a good idea to avoid vine stress, and there are different things that can be done. For example, compost can be applied, and there are also biocontrol agents that we are evaluating. So there are a few other things that can be done, and this is after planting. So the number one is avoid any type of injuries, because that's where the infection occurs. Late pruning to delay bud break can be done. Irrigation water and fertilization should be managed, especially late in the season, to avoid like a lot of vegetative growth late in the season. And then hilling up, that's something that's been done a lot in Ontario, not so much here in BC. And that involves hilling up the soil around the graft union in fall. So this photo here, for example, shows that the soil for this vine was hilled up until this arrow. And no galls have developed in the bottom trunk because the soil was insulating the trunk. And there have been studies that show that the temperature below the soil is about 5 degrees warmer than if the trunk would be exposed to the air. However, in this case, some galls still developed higher up on the vine, but the lower part was protected. And then multiple trunks, that's also something that can be done. When the main trunk is affected, you can train some of the other shoots to replace the main trunk. And then in general, just monitor and replace any vines that are infected. And if you do that, make sure to remove as much of the roots as you can, because we know that the bacteria can stay in the soil. So what you are doing, like whether training multiple trunks that has been successful here in a vineyard, or removing or replacing, that probably depends on your individual situation. So now we're moving in some of the work that we have done in our lab. So the first thing we have done is working on early detection. So we've tried to develop a methodology that allows us to measure the abundance of the bacteria in different plant materials. So we have used gall material, we looked at vineyard soil, but we've also taken roots from dormant nursery stock before they were planted in the vineyard. And we did isolate DNA. And then we used a method called droplet digital PCR that quantifies bacteria. So using this method, we can actually say how many bacteria we have in a sample. This method is a lot more sensitive than some of the other methods that are being around. And it doesn't matter if there are any soil contaminants or phenolics that came in from the plant. So that method will still give accurate measurements. And the method is very fast, because previously we had to isolate bacteria and plate and then wait for them to grow and identify. So now we can do everything from isolation to having the results in one day. So we use this methodology in a nursery study, and this has been a collaboration with Jose's lab in Summerland. They have collected nursery material from different domestic and international grapevine nurseries, and the material came in from different years, but it was mostly Chardonnay, Merlot, Pinot Noir, and it was on the same rootstock. And Jose's lab has divided the nursery material in 4 different plant sections. So the roots, the base, the graft union, and the scion. And they then isolated DNA that we used in our ddPCR methodology to quantify the bacteria that we found. In addition to the material that we received from Jose, we've also had some of our own samples, and mostly root samples that we've analyzed as well using the same method. So this is our first result of the study. This column shows you the different nurseries that we've looked at. So nursery A, for example, we got samples from 2014, 18, and 19. Those are all the cultivars that we've sampled. Not that many different rootstock, but a few. And here is shown the incidence of the bacteria. So by incidence here, I mean, this number shows the percentage of the plants that harbor the bacteria in at least one plant section. So if we found bacteria in the roots of the plant, maybe not in the cyan, this still counts as positive. And those numbers here, percentages, are really high. So most of them, the whole batch that we received, 100% incidence. So we found bacteria in all of the plants. There was just one nursery here, and that's actually was a local winery, and they have done their own cuttings from a vineyard. So these are very, very low. All the others that came in from nurseries had very, very high incidence. So the bacterium is present in all of the nurseries that we've sampled and in all of the years that we've looked at. So that's very concerning. However, if most of the dormant nursery material that we receive here into BC is infected, why aren't all vineyards diseased? I mean, there are many, many vineyards that do not have any crown gall symptoms. So this depends on two conditions, on two factors. The first one are environmental conditions. So only when there's wounding will there be disease. So if there's no wounding, no freeze injury, we may not see any disease establishing. And the second is the threshold. We don't know how many bacteria we need in order to cause disease. So is it enough to develop a disease if we find 10 bacteria in the plant, or do we need 100,000 bacteria? So in order to find that out, we did a greenhouse assay where we inoculated grapevines with different concentrations of the bacteria. So that ranged from 5 bacteria to 500,000. And we inoculated them by a needle prick inoculation right into the tissue. And we found that after 7 weeks, 2 out of the 5 replicates developed galls when they were inoculated with 5,000 bacteria. All of the 5 plants developed galls at 50,000 bacteria. So we believe that the threshold in order to develop disease lies somewhere between 5 and 50,000 bacteria. And this is a very high number. And this is the reason why hot water treatment from the nurseries may still be an effective treatment. Because if we can reduce the number of bacteria, even though if we can't eliminate them completely, but if we reduce them, the vines may still make it over the first years. And if they reached maybe like 3 or 4 years, they may have overcome and they'll be fine, even though there is a virus present. So back to the nursery study, this graph shows the root data, so only the root samples from the different nurseries. And interestingly here, for example, nursery C in 2014 and in 2018 had high numbers of bacteria, but then the same nursery in 2019 had very low numbers of bacteria, even though this is the same cultivars, the same rootstock. So what that tells us is that we can't generalize. Like when we receive material 1 year from a nursery and it is infected, it doesn't mean that in the next year we'll have the same problems in the vineyard. Differences in the propagation process may have made between those years, or the material is coming from different mother blocks or different source material has been used. This is the nursery data, again, now separated by 4 different nurseries. And within each nursery, I'm showing the different cultivars and then the abundance of the bacteria at the different locations on each vine. So as a take-home message here, there is no pattern. Like we don't see any location that consistently had the highest numbers or any cultivar that consistently was higher or lower than the others. For example, nursery B, cyan had the highest numbers in the Chardonnay, but in nursery E, it was roots. So, and this is problematic for our testing because usually we take root samples before testing because we can only take root samples because the plants need to be viable for planting. So we can't cut off the pieces of the cyan. But if we don't see a virus in the roots, then based on this data, this doesn't mean that there are no bacteria at other places in the vine. And by only looking at roots, we may underestimate the infection. And then what this also tells us is that the bacteria are non-uniformly distributed. So we don't know They can be in any section, like either the root space, graft site, or anywhere in between. The fact that bacteria are non-uniformly distributed in canes has been shown previously. And this is a study where they've looked at 10 canes here. Those are numbered 1 to 10. And then here is a location on each cane. So node 1, internode 1, and so on. So if they would have sampled cane number 4, for example, at internode 1, they would have said this plant is crown gall free. However, bacteria are located at other places. So you have to basically sample the whole vine to have like a good idea of where the bacteria are located. This study also found that bacteria are not only present in the canes, but also in the green shoots, and they are located outside on the leaves. And they also found that bacteria are in wild grapes. So as conclusions for the nursery study, we find that A. vitis is likely present in most of the grapevine nurseries, at least the ones that we have studied here. But the expression of the symptoms depends on 2 other factors. First, there need to be enough bacteria present, and then the conditions need to be right. So wounding needs to occur. The threshold that we found is pretty high, and the bacteria are non-uniformly distributed throughout the plants, which has implications for the testing. So if we only continue to do root sampling, then we may underestimate the bacterial abundance. What we don't know, though, based on this study, is where the bacteria are coming from in the nursery. So is it the infield, is it the propagation process, or mother blocks? And that's something that we're going to have to follow up. I know for trunk diseases, for example, sources of infection in the nursery are known, but we don't have this information for crown gall disease yet. Now we're moving on to our compost study. There are many benefits of compost. It's a sustainable management strategy. It does improve soil fertility. And in other studies, it has been shown to increase yield, improve crop quality, and help with water retention. And there are many studies that show that compost suppress soil-borne diseases, and that's mostly true for fungal diseases. So our rationale was that if an increased organic matter through application of compost could maybe diversify microbial populations, and then we may have other microbes that can act as antagonists against A. vitis and keep the population down. In addition to that, there was a study from Tom Forge, who found that compost treatments did suppress nematodes, because there is a connection between nematodes and A. vitis, which was shown in the study here, where they have inoculated soil with A. vitis only, and then planted vines, and no galls have developed. But then they used A. vitis and nematodes into the soil, planted vines, and they found that many galls have developed in those vines. And when they've used nematodes only in the soil and planted vines, only small galls form. So this again supports that nematodes do actually help A. vitis to enter roots of grapevines. So this study was done in a local vineyard in a Chardonnay that was established in 2014. And this site has previously been a pasture. So there was no grapes before. So the symptoms that we've seen here in this vineyard, they all came in through infected nursery material. The study ran from 2019 to 2022, and we had 4 treatments. So 3 different composts. Compost 1 was yard waste. Compost 2 was waste from a commercial winery, and compost 3 was yard waste, but some peat was mixed in. So this is how we laid out the plots. One plot consisted of 5 vines, and the outer 2 vines were guard vines, and the inner 3 were treatment vines. So we've only taken our samples from the inner 3 vines. And the compost was applied every year in spring on the surface, and we did normalize that to organic matter content. And we've looked at a lot of different measurements. So we've looked at plant performance, specifically grapevine phenology, like bud break and bloom. We've looked at leaf greenness, because that can be a measure of stressed vines. We counted clusters, assessed yield and pruning weight, And then in fall, we've looked at the effects of compost on berry quality. So we did assess Brix, TA, pH, and berry weight. And we've also analyzed the soil every year. And then most importantly, we've looked at disease severity. So we did visually rate all of the trunks for the percentage of galls. Then we did quantify the bacteria in the soil. And then Tom and his team did nematode population counts twice a year. They also helped with application of the compost and taking all the soil samples. So here are the results of the compost study. Unfortunately, there was no difference in crown gall disease incidence between any of the treatments. So there was crown gall, but compost didn't have any effects. And then there was very few cells of avitis that we found in the soil. And that's very interesting because there have been many bacteria before, and now there are very few. But this wasn't because of compost, because we don't see a treatment effect. So something else must have happened in that vineyard that reduces the population of the bacteria. There are some nematode results though, and I'm not an expert in this, so please ask all nematode questions to Tom. Because this vineyard was newly established, there were very few nematode populations there to begin with, but Tom found dagger, pin, root lesion, and ring nematodes. So as the vineyard grew over time, dagger and pin nematodes increased as well because they do parasitize grapes. Root lesion nematodes do not parasitize grapes, and their population stayed low. However, ring nematodes do parasite grapes, but their population was also low. So something else is going on in these vineyards that's suppressing the population of these nematodes. And I believe Tom is going to look into this further. However, there was an effect of compost in this experiment. So the dagger nematode population was lower in the yard waste compost. There were a few other smaller results. So for example, leaf greenness was increased in the peat compost. That means that the nitrogen content in those vines were a little higher than in the others, but the differences were really very small, and we don't believe that there is a real biological significance to that data. Then yard waste and peat compost increased berry pH, but the pH of all of the treatments was in the optimal range for winemaking. So again, this increase really isn't biologically significant. And then the other effect was that the winery waste compost has increased berry weight, and that's more of a negative effect because we don't want to dilute berries with increased weight. There were good results though if we were looking at the percent nitrogen in blue and carbon in orange and organic matter in gray. All of the three composts, all of the three measurements were increased towards the control. And that is an indicator for improved soil health because a high organic matter content supports diverse soil microbial communities, and that can suppress pathogens. And then high organic matter content can also improve soil structure, water storage and movement, and soil fertility. So this was a good outcome of this study. So as conclusion for the compost study, unfortunately, compost had no effect on crown gall, but we did see that yard waste compost did suppress the dagger nematode populations. Compost increased leaf greenness, berry weight, and pH, but likely this is practically not very significant. But compost did improve soil health by an increase of the soil nitrogen, carbon, and organic matter. Now we're moving to our Healing Up study, and that's something that has been done frequently in Ontario, and it has been very successful there. So this was a vineyard that was established in 2014, also in grassland. So again, all the disease that we have been seeing in this vineyard came in through the infected nursery material. In 2017, about 30% of the vineyard was infected with crown gall, and about 20% were dead. So at that time, we did an initial soil sampling, and we found about 6,000 bacteria per gram of soil. In 2020, we've taken out vines, always in sets of 3, and those vines were dead. So those were removed, and we planted new vines. And again, we had guard vines on the outside and a treatment vine in the middle. And in fall of the years of 20, 21, and 22, we healed up the soil, and we did that just by moving the soil up to cover the graft union with the hand. And then in spring, the soil was removed again. And we rated for crown gall in summer of 22. And we found that 3 of the plants that did not have soil healed up, so that were unprotected, had galls that developed. But we did not find any galls in any of the plants that were healed up. So I have to say that we only had 8 replicates in total. So this is 3 out of 8. And this is a low number, but no galls in any of the healed up vines. We also did sample the soil in fall of 2022. And surprisingly, we did not find any Avitis bacteria in the soil, or like really, really low numbers compared to the high number of bacteria that we had in 2017. So over the course of these 5 years, again, something has happened in the vineyard that reduced the agrobacterium soil population. So the conclusion here is healing up prevents gall formation, and that's in parentheses because it is only one site we've looked at, and it's a low number of replications. And the bacteria did not survive in soil after 5 years. In Ontario, there were problems though associated with healing up, because when it's done commercially, a lot of machinery is being used. And that machinery has been adding some wounding to the plants. So they had some vineyards where there were actually more crown gall symptoms in plants that were healed up. And it was only because of the wounding caused by the machinery. So this is my last study here about biocontrols. So in general, biocontrols is a method to control plant diseases by using other organisms. And for example, crown gall of stone fruit trees can be controlled by biocontrols. And that's what I've spoken out before. This is the strain K84 that's contained in those products, but they don't work for crown gall of grapevines. There is a paste that's called Gall-X that can be applied on the trunk after the galls are removed. But the bacteria are systemically inside the vine. So Gall-X can't reach inside the vine. It can only protect the site where the gall has been, but it will help something there. So there are many biocontrol studies available, and mostly the ones from Tom Burr and Akira Kawaguchi are the ones that are most advanced. And there are some considerations for biocontrols. So we need to be able to culture them in the lab. So they need to be able to grow with us in the lab so that we can work with them. Biocontrols need to also grow into the plant environment. So depending on how we'll apply that later in a product, they need to be able to grow in xylem where there is another pH compared to the soil, or they need maybe to be able to colonize the roots depending on application. The biocontrols also need to grow in a local environment. And that means that maybe a biocontrol that was isolated in South Africa may not be working here in the Okanagan Valley. And biocontrols also need to be effective against local Avitis strains, because the pathogen strains that we have here in the Okanagan, they are very likely different from strains in Oregon or any other areas. And when we started this project, we didn't have any locally isolated pathogens. So we did a survey and we sampled many vineyards throughout the Okanagan Valley. And we now have a collection of isolates. So all of these are isolates that we now have available. They have all been sequenced, so we know they are Rhizobium vitis. And the first 6 have been inoculated into tomato and grapes, and we were able to generate symptoms. So we know that they are virulent. In addition, all of these samples have been sent to Dr. Alexandra Weisberg from Oregon State University, and she was able to sequence the genome from all of those strains. So now back to the isolation of biocontrols. So we have obtained a lot of roots from BC and Ontario vineyards. Those roots were washed, and then the solution was pipetted onto plates. And all of these bacteria were then isolated, and that resulted in rhizosphere bacteria. So those bacteria were associated to the outside of the roots. The roots were then ground up and plated again And this allowed us to isolate endophytes. So those are bacteria that reside inside the plant. So now we wanted to test our bacterial collection, and we did that by conducting a plate inhibition assay. And that's the schematic here. So that's the plate. And we did plate the biocontrols in the middle, and then the locally isolated Vitis strain, so the pathogen on the outside. And we did several rounds of tests. We changed the plating order. We did different mediums. We used different type of pathogens, and that allowed us to reduce our population or our collection to about 5 isolates that have been worked very well. So we did sequence those, and mostly they were identified as Bacillus and Pseudomonas. And this figure here shows the results of inhibition on the plate. So again, here is the biocontrol, and this is the pathogen. And here we see a weak inhibition because the pathogen was plated all the way on the red line, but it doesn't grow here because something of the biocontrol inhibited its growth. And on this plate, we can see complete inhibition. So the pathogen was not able to grow at all because of the biocontrol being here. So the next step was then to move these experiments into plants and doing a needle prick assay. So we have grown the biocontrol and the pathogens and mixed them 1-in-1 and inoculated plants. So we pipetted 5 microliter of that solution onto canes, and we then stuck a needle into the drop, and then the negative sign and pressure takes that drop in, and bacteria disperse in the plant. The needle is what has caused the wounding in this case. So 8 weeks after, we found galls that have developed, but they developed on all of the plants. So there were no differences of the biocontrol. So biocontrol wasn't able to reduce gall formation. We then changed up the experiment a little, and that we used double the amount of biocontrols and inoculated them first, waited 24 hours for the bacteria to establish, and then added 1 part of the pathogen, again with needle prick, and waited another 8 weeks. And again, galls did develop, but again, there were no differences in size of galls. So the biocontrols that we have looked at here did so far not inhibit crown gall formation when we've used this type of method. But that's not really how it's working in the field. So we are not using needle prick inoculation. So we have then changed our method and evaluated an in-planter root soak assay. So for this, we've obtained nursery material. We pruned the roots and then pruned the roots and soaked the plants in a biocontrol solution, and then planted those nursery material in soil that has contained the bacteria. We then wounded the stem, and then we waited 9 months, and there were no galls. So that assay doesn't work in the greenhouse, whereas in the field, we plant nursery material in a vineyard that has crown gall bacteria. Those vines get sick, but somehow we are not able to replicate that in a greenhouse. So we changed up our experiment, and instead of nursery material, we are using grapevine shoots that we've obtained from a vineyard here. We grew roots on those shoots, pruned those roots, and soaked again in the biocontrol and pathogen solution, planted, and yay, we found 1 gall. But of course, we can't say anything about the effects of biocontrol if we only come up with 1 gall. So we now changed our assays to using tomatoes instead, because tomatoes can be used as an experimental host. They grow a lot faster. We don't have to wait 9 months for symptoms to show up. Symptoms usually show up after like 7 or 8 weeks. So same assay, planted, and there are galls right now, and on many plants, but unfortunately it's still ongoing. So we can't say if any of the biocontrols have any effects in planter so far. So far I've been talking about bacterial biocontrols, but there are also fungal biocontrols available. For example, Trichoderma. This is a fungal genus, and it's being found in many soils. And there are a lot of reports about Trichoderma as a biocontrol against fungal pathogens. So we repeated our in vitro assay with some of those isolates, and we've obtained all of those isolates from Jose's lab. And those are the results. So we do have inhibition on place using Trichoderma. And interestingly, that two of those are also working very well for trunk diseases. So we are hoping to do more experiments there and work together with Jose to find out if we can use those trichoderma strains to maybe control both trichoderma and avitis. And also implanter assays are still ongoing, where we've used trichoderma for plants. But then we've seen trichoderma works on plates, and there already is a product available that has trichoderma strains as active ingredients. So we wanted to try out if this product, because it's already available, has an effect on crown gall. We did that by vacuum infiltration, and Jared helped us a lot with this method here. So we used canes, and they were hooked up to this vacuum apparatus, and the canes were dipped into a solution of root shield. The vacuum was applied, and then the solution was distributed throughout the cane. 10 days after this infiltration, we then just confirmed that the fungus was indeed colonizing the whole cane, and yes, it did. We then rooted the canes, planted them in the greenhouse, and then repeated our needle prick inoculation assay. But unfortunately, galls did develop, but there was no difference in gall size. So the root shield didn't have an effect when we used it for vacuum infiltration. We've also looked at pruning weight, and there was no effect of root shield in terms of pruning weight. So conclusion for the biocontrol study, we did identify bacterial and fungal isolates that do inhibit avitis, but so far we are only successful in the lab. Root shield wasn't effective when infiltrated, but we have experiments ongoing now where we're using root shield to colonize the roots, and maybe the trichoderma colonizing the roots can prevent avitis bacteria to enter the roots from the soil, but those assays are still ongoing. And then our implanter assays are not reliable so far, and we're not the only one. That's most of the studies report biocontrols in vitro assays, but very few have actually results available from implanter assays. So we're wanting to look at a seedling assay, because that's one that's been reported as successful. So as a summary for all of the research that we've done, we do have a quantification method available that we can use to test soil and plants before planting. We do have local avitis isolates available, and we have the genome sequenced. There is a high threshold for gall formation in nursery material, but the bacteria are present in most of the nurseries, or in all of the nurseries that we've sampled in this study. We do know that the bacteria are non-uniformly distributed, with implications for the testing. Compost didn't have an effect on crown gall incidence, but it did improve soil health. So far in our study, we've seen that the bacteria do not survive in the soil for more than 5 years. And we do have potential bacterial and fungal biocontrols available. And healing up in this, our study site did prevent gall formation. So for the next round of projects, I'm hoping to do some more research. One thing we want to do is look at the nursery materials. So we want to know where in the process the infection occurs. So is it the primary material? Is it the propagation process? Is it the mother blocks or the infield study? In addition, there are nematode-resistant rootstocks available through UC Davis, and we want to look at those and test them for susceptibility to avitis. We also want to dig deeper in how long avitis survives in the soil. We don't know what happens within these 5 years. So this would be very interesting, because that can tell you how long you'd have to leave your vineyard fallow, so the population can decrease, so you can plant vines and they won't have disease. We want to expand on healing up experiments. So here I'm putting a call out to growers. If you are planting a vineyard next year, and it is in an area that's prone to frost injury, or you even had crown gall in your vineyard that you're replanting, please let me know, and I would love to come and do some more experiments with the healing up. The most promising strategy, I think, is going to be biocontrol. And we do have collaborators And one of the collaborators has a strain that is now within a company. So they are working on developing a product. And once the new project starts, we will be able to bring this product and try this out here locally and see if it works against our local A. vitis strains. Another thing we want to do is isolate bacteria from wild grapes, because we do know A. vitis is present, but wild grapes don't seem to have symptoms. So maybe there is something there within the wild grapes that protects them. We also want to do more trichoderma research, and we have to do more implanter testing in general, but for that, we're going to have to develop our seedling assay. So this is it for my presentation. I'd like to thank you everybody for listening. And I specifically want to thank Portia, our PhD student. So she did most of the work that you've been seeing here. Louise Nelson and her lab, they helped a lot. And then big thank you to Quails Gate, and specifically Chad, Tim, Judy, and Jordan. It was great working with you. Thanks a lot to Tom and Paige, who helped with the compost application, the sampling, and did all of the nematode work. Then Jose's lab has provided a lot of information and strains and help throughout the years. Carl has been doing our berry analysis. Wendy from Brock shipped some of the root samples from Ontario. And then Lynn Bremer also did some of the berry analysis for us. And I want to thank Hans for answering all of my questions, and Melody and Alexandra from Oregon State University for sequencing our strains. And then I want to thank all of the wineries for allowing us to visit your vineyards, for sending in samples. And yeah, it was great to be able to work with everyone. Thank you.
Thank you, Tanja. We already have a question in the chat from Danielle. How does humidity Okay, Zoom, thank you for that. Sorry. How does humidity accelerate crown gall prevalence? I'm comparing a BC experience to a Nova Scotia experience. Crown gall acceleration on the east coast of Canada is extremely destructive and fast by comparison to the Okanagan Valley. Any insight as to why?
Yeah, that's something that has been observed in California as well. They don't have a lot of freeze injury because it's not cold, but they have seen an increase in crown gall as well, and they attribute this to humidity. So I'm not sure if humidity can cause wounding on vines, but I'm not entirely sure how humidity affects vines. But that's something that's not only, yeah, that's also been observed in California.
Is it being studied?
I haven't seen any studies about that interaction, no.
Yeah. Hans is asking, how fast do A. vitis bacterial counts increase on plants with relatively low initial numbers? Would this increase be linear, or are there factors that can speed up or slow down multiplication? Do old plants over 20 years infected from the propagating material used eventually have declining counts?
So there are no studies tracking the count in the vine so far. But we believe that a vine can overcome bacteria like in numbers. So with the age, that the vine will get stronger and can actually fight bacteria. Sorry. It depends on wounding, right? So if there's no wounding in the plant, then bacteria that wouldn't multiply. So wounding is what's creating the population to increase. So if there's no wounding happening in those plants, and maybe they can have low numbers of bacteria for a long time. But it would be an interesting study subject to find out, like, over time. There's also seasonal changes. There have been, for example, more bacteria found in the spring and in the fall versus in the summer. But we don't know exactly why that happens. that may have to do also with the sampling location.
So with an older vine, like a 20-year-old vine, the trunk would be more robust, like less susceptible to wounding. Is that true?
I don't know the wounding, but I think yeah, we see most of the like freeze injury, for example, are more prevalent in some of the younger vines. So because of that, they would be more susceptible. But there's few agrobacterium that I see here in the Okanagan in the older vines. It's mostly the younger plantings.
What about cane versus spur pruning?
So far there is no study looking at that. That would be something interesting to look at, like if pruning has an effect. But then the bacteria, again, they can be anywhere. So they can be in the canes or the trunk or in the roots. So and in spring they could colonize all parts again coming from the roots.
Any other questions? I mean, I have some questions, so.
Yeah, I'm sorry, there's nothing really concrete that we can do right now. And then I know there's a lot of worry right now because of the winter that we had and all the damage that has been done.
So can you explain, so you mentioned earlier that one of the things you can do is delay pruning or late pruning to delay bud break. Can you explain how that works?
Yeah, that's just to prevent damages in the buds. So if you delay it, then there's less risk of damage later on.
From cold?
If there would be a frost event in spring. Okay. Yeah.
Okay. And then also you talked about removing and replacing the trunks. So that would be on the same root system, right?
No, you would be taking out the whole vine with the roots.
Okay.
Hopefully.
So
yeah. And then try to take as much of the root system out because they can stay. Like if there is any leftover debris from the roots, the bacteria can stay associated with the plant material there. And then if you plant new vines, then they can get infected. So that's something that we want to look at. Do a detailed study of how long they can actually stay in vineyard soil. Because right now we only know 2 years, but it looks like in 5 years they're not in the soil anymore, so somewhere in between.
And do you know like the distance? Like what area would you have to remove around the infected plants and then wait 5 years?
So they're located in close association with the plant roots. So you wouldn't have to remove all the soil. So if you can get out most of the plant material, that would reduce the populations. So the soil would attach to those plant roots. So it's something that's immediately attached to the roots. Okay,
so just the radius of that plant, sort of.
Yeah, and I know this is very difficult to do because the roots go very deep and they spread. So.
Chris Mark has a question. Are there any remote ways of mapping crown gall to allow tracking in larger vineyards?
So far, we don't. I mean, I know there's GPS work being done to monitor vineyards. Symptoms, I guess you could track water stress symptoms, but then there may be other causes for that. So it doesn't necessarily has to be crown gall.
Right. Okay. Right, we're just about out of time, so I think there are no more questions. I think that this is a good year for people to be really having an eye on their vineyard after the winter that we've had, to see if the damage has invited crown gall development. And can they, you gave your email. If people have further questions, they can email you, and also Kate will put it up on our website, and this presentation will be available probably later today on the website. Thanks so much for joining us, Tanja.
Hey, thanks for inviting me.
Yeah, I hope to see, further developments.
Yeah, thank you.
And thanks everyone else for joining us. We'll see you next third Thursday. It'll be June 15th. And we'll have a presentation from Chris Marks. Thank you. Bye-bye.
Bye.